Welcome back guys. Here in this video let's discuss about cell membrane. Guys, here you can clearly see cell membrane with different components. The ones which you are seeing in blue color, that layer is made up of lipids which is arranged in two layers. So lipids are arranged in bilayers. Okay, there is outer leaflet as well as inner leaflet. Bilipid layer is seen, in which you can also appreciate there are carbohydrates, proteins. So we can say cell membrane is something made up of lipids, carbohydrates as well as proteins. So let's write it down here. Components. What are the components of the cell membrane? The first component is proteins. Second component is lipids and carbs. Okay, now why I have taken this order? Because proteins are accounting almost 55% of the cell membrane. Lipids are accounting for 40% in the cell membrane, and carbohydrates are accounting for the least, only 5%. So the majority of the cell membrane, the most percentage in the cell membrane is taken by the proteins. Okay, but important exception which you need to know is nerve cell membrane. Okay, now what is the exception here? In the nerve cell membrane, proteins are only contributing to 20% and lipids are almost 80%. Okay, usually proteins are the ones which should be the major component, but here proteins are only accounting for 20%. This is what you need to know. Now let's discuss about fluid mosaic model. Fluid mosaic model. First of all, who proposed this fluid mosaic model? Fluid mosaic model was proposed by Singer and Nicholson. Okay, fluid mosaic model was proposed by Singer and Nicholson. Here the fluid at the C is represented by lipids. I have already taught you lipids are arranged in bilayer. Okay, there is a bilayer fluid or the sea of lipids. There is a sea of lipids in which the proteins are floating. So mosaic represent proteins. Okay, so in the sea of lipids, proteins are floating. That's what is called as fluid mosaic model given by Singer and Nicholson. Now let's discuss. I have already taught you that proteins are the major component. Just know this, proteins are the major component. Okay, one important MCQ which you need to know for your FMG exam is that major percentage or the major concentration, highest concentration of proteins is present in which cell membrane? Yes, we know proteins are accounting for 55% of the cell membrane, but even greater than that, proteins are present in one of the cell membrane. Which cell membrane? In the inner mitochondrial membrane. So highest concentration of proteins are present in which cell membrane? Is present in inner mitochondrial membrane. Okay, that's a MCQ which you need to know. Now after this, let's start with the lipids. Okay, we have seen lipids are arranged in bilayer fashion and the lipids are just like a sea. Let's classify the different types of lipids in the cell membrane. The lipids are majorly classified into three types, or we can say three different types of lipids are present in the cell membrane. First type of lipids are called as phospholipids. Okay, lipids with the phosphate groups. And second type are Glycolipids. Here very simple, lipids means fats and the glyco means the carbohydrates. So lipids are associated with some carbohydrates, and the third type of lipids are sterols. Okay, with the sterol rings, lipids with the sterol rings. So three types of lipids are phospholipids, glycolipids as well as sterols. Now, what are the different types of phospholipids which are present in the cell membrane? The first type of phospholipid, which is very important, is phosphatidylcholine. Okay, phosphatidylcholine, which is also known as lecithin. The second type of phospholipid is sphingomyelin or sphingomyelin. Okay, so phosphatidylcholine or lecithin. Second type of phospholipid is sphingomyelin, and the third type of lipid is phosphatidylserine. Okay, guys, please concentrate. Here I am telling you phosphatidyl. See, that phosphatidyl is representing that phosphate group. Okay, so phosphatidylcholine, phosphatidylserine, and one more thing is there, that is phosphatidylinositol. And the last type is cardiolipin. Don't worry, we will discuss in detail about all the important points about these different types of phospholipids. Now here I am showing you the classification. Lipids are majorly classified into phospholipids, glycolipids and sterol. I have shown you different types of phospholipids. After that, now let's discuss about the two important types of glycolipids. The first important type of glycolipid is cerebrosides and gangliosides. Okay, cerebrosides and gangliosides, and sterols include cholesterol. Okay, cholesterol is the sterol. So this is a broad classification of different types of lipids. Here for your exams, I just want you to know this cardiolipin is there, right? This cardiolipin. Now there are certain diseases where the person is going to produce antibodies against this cardiolipin component or cardiolipin phospholipid. Now we can say these antibodies as anti-cardiolipin antibodies, anti-cardiolipin antibodies. So what are those diseases where you can see anti-cardiolipin antibodies? Examples include two important such diseases are syphilis. Could you able to remember the syphilis is a sexually transmitted disease caused by which organism? Treponema pallidum, a spirochete, which can be seen in dark field microscopy. Okay, these are important points. And these cardiolipin antibodies are also seen in one more condition called as APLA syndrome. APLA. What exactly is this APLA syndrome? APLA syndrome is anti-phospholipid antibodies. Yes, cardiolipin is also a phospholipid, right? So anti-phospholipid antibodies are nothing but anti-cardiolipin antibodies, okay, which will be causing recurrent abortions in females, that you will study in your pathology or gynecology. But here I want you to know that anti-cardiolipin antibodies are seen in two conditions. One is syphilis and second one is APLA syndrome, that is anti-phospholipid antibody syndrome. Now after saying this, let's discuss one by one. Okay, first let's discuss about phosphatidylcholine. Let's write it down here, guys. Phosphatidylcholine. Okay. Now, what are the important points which you need to know for your exams regarding phosphatidylcholine? See, this phosphatidylcholine, the full name is dipalmitoyl phosphatidylcholine. Okay, dipalmitoyl phosphatidylcholine, also called as lecithin. Okay, I have already taught you this. Now, this lecithin, it's a major component, or I should say major lipid in something called as surfactant. Okay, now what exactly is the surfactant? See, surfactant, it's a lipoprotein which is present in the alveolus. Okay, usually in the alveoli there is something called a surfactant. Now what is the function of surfactant? Surfactant is going to reduce the surface tension. Okay, see, all these phenomena, all these things were in detail discussed in the respiratory physiology, but here also I am just giving you a glimpse. Surfactant is present in alveolus. Now the function of the surfactant is to decrease the surface tension. Actually, surface tension is a very bad force. It's a collapsing force. It will cause collapsing of the alveoli, which is not good. So in the alveolus, to prevent the collapse, there is something called a surfactant. Now the surfactant is a lipoprotein. Okay, surfactant is a lipoprotein. So it's a lipid as well as a protein. Now what is that lipid? See guys, I am again writing down here so that it will be easy for you. Surfactant. Now the surfactant I have taught you, it's a lipoprotein, which means it's made up of lipids as well as proteins. Now these lipids are further divided into major lipids and minor lipids. Okay, now what is the major lipid? Phosphatidylcholine, our lecithin. Okay, our lecithin is the major lipid in the surfactant. Then who is the minor lipid? Don't worry, in the next upcoming slides we will discuss who is that minor lipid. Okay, so the major lipid is nothing but dipalmityl phosphatidylcholine or lecithin. Okay, now apart from this, what else you should know? Deficiency of this palmitylcholine, okay, phosphatidylcholine, it will cause which disease? Deficiency of dipalmityl phosphatidylcholine or lecithin will cause a disease called as hyaline membrane disease. Okay, hyaline membrane disease in the newborn. Okay, it causes hyaline membrane disease in newborn. See, it's very simple. In the newborn, there is deficiency of this lecithin. Whenever there is no lecithin, there is no surfactant. Whenever there is no surfactant, the surface tension forces are very high. Now such high surface tension forces will cause collapsing of the lung, causes the distress in the baby. So that is called as hyaline membrane disease or respiratory distress syndrome. Now after this, let's talk about sphingomyelin. Okay, we have discussed about the lecithin. Now let's start with the sphingomyelin, guys. Sphingomyelin. What are the important points which you need to know regarding sphingomyelin is, see, we have discussed who is the major lipid. The major lipid is dipalmityl phosphatidylcholine. Now the minor lipid in surfactant is sphingomyelin. Okay, now this is the MCQ. Now this sphingomyelin, yes, it's a minor lipid. And the one more important point is lecithin sphingomyelin ratio. Greater than 2 indicates fetal lung maturity. Okay, this is a very important point which you need to know. Normally, who is the deciding factor for the maturity of the lungs in the baby? It's a production of surfactant. See, in a newborn baby, or I should say in a fetus, and the baby is in the uterus, the lungs in this baby are non-functional. The lungs in the baby are not matured. Somewhere around 28 to 32 weeks, the alveolar cell type 2 pneumocytes in the lungs, they will produce something called a surfactant. Now this surfactant is the one which makes the fetal lungs to mature. Now what I am saying, surfactant is nothing but lecithin as well as sphingomyelin. So whenever you have enough amount of lecithin and sphingomyelin, enough amount means whenever the ratio is coming greater than 2. Whenever the ratio between lecithin and sphingomyelin is greater than 2, it indicates yes, there is enough amount of surfactant in the fetal lungs. Now we can say that the fetal lungs are getting matured enough. Surfactant is there. It is decreasing the surface tension. Okay, so lecithin sphingomyelin ratio greater than 2 is an indication of fetal lung maturity, and also surfactant is somewhere produced around 28 to 32 weeks, and surfactant is made up of lipids as well as proteins. What are the two important lipids? Lecithin as well as sphingomyelin. And what is that protein? Don't worry, we will discuss. That protein is nothing but called as surfactant protein. Okay. Now after sphingomyelin, let's discuss about phosphatidylserine. Okay. Let me write it down here so that it will be easy for you. Phosphatidylserine. Okay. Now usually this phosphatidylserine is something expressed inside the cell or towards the cytoplasm. Okay. See guys, in a healthy cell, you can see these are phosphatidylserine molecules which are towards the cytoplasm, not towards the outer side. They are expressed towards the inside in a healthy cell. But whenever a cell is damaged or whenever the cell is undergoing apoptosis, now there is a flipping of this phosphatidylserine. See here, it's very clearly given phosphatidylserine is now flipped towards the outer side. Okay. Now, how this is important to us? If you want to know whether the cell is healthy or whether the cell is undergoing apoptosis, there is a marker called as annexin 5. Normally, if you put annexin 5, okay, whenever you put annexin 5 onto a healthy cell, this annexin 5 cannot bind to phosphatidylserine because phosphatidylserine is something is there towards or inside towards the cytoplasm. So, in a healthy cell, this annexin 5 molecules, they cannot bind to the phosphatidylserine. But when a cell is undergoing apoptosis, these phosphatidylserine molecules, they are expressed outside. So the annexin molecules, our annexin molecules, these dyes, they will go and bind to the phosphatidylserine. Now they will emit the fluorescence. So we can clearly say annexin 5 is a marker of apoptosis. Okay, so annexin 5 is a marker of apoptosis, and also know phosphatidylserine in a healthy cell is expressed inside towards the cytoplasm, not outside. Now after this, let's discuss about phosphatidylinositol. Okay, let me write it down here. Phosphatidylinositol. So what are the important points which you need to know? This phosphatidylinositol is also called as inositol triphosphate. Okay, written as IP3, inositol triphosphate. Now it acts as this IP3 molecule, it's acting as a secondary messenger. Now what are these secondary messengers, primary messengers? We will discuss in the topic of cellular messengers. Okay, that's a totally different topic, but for now just trust me, this inositol triphosphate is something acting as secondary messengers. Whenever it's activated, it causes calcium influx into the cell, which means it allows the entry of calcium from outside to inside. So calcium influx, it allows calcium to flow into the cell. So inositol triphosphate, okay, inositol triphosphate is other name for phosphatidylinositol. It's acting as a secondary messenger. So you all guys have studied in your class 11th and 12th the secondary messengers like CAMP, IP3, DAG, diacylglycerol. So remember, secondary messengers include CAMP, cyclic AMP, inositol triphosphate, IP3, and diacylglycerol, DAG. Okay, these are all the secondary messengers which we'll discuss in detail in the separate lecture. But for now, just remember IP3 inositol triphosphate is a secondary messenger, causes calcium influx. Now after this, let's discuss about cardiolipin. Okay, now already we have seen anti-cardiolipin antibodies are seen in conditions like Apla syndrome as well as syphilis. Now this cardiolipin, yes, it's present in the cell membrane, it's a phospholipid, but maximum or abundant amount of this cardiolipin is present in the mitochondria. Mitochondrial membrane, okay, mitochondrial membrane of nerve cells. Okay, this is an important point. Cardiolipin is abundant in the mitochondria of nerve cells, and anti-cardiolipin antibodies are seen in syphilis as well as Apla syndrome, and also they are seen in lupus also. Okay, just let me write it down here for the completion purpose. Antibodies, okay, anti-cardiolipin antibodies are seen in syphilis, Apla syndrome as well as SLE, systemic lupus erythematosus. Okay, just know at least these 2 points. Now after this, let's discuss about the glycolipids. What are the 2 important type of glycolipids, guys? We have discussed. There are gangliosides as well as cerebrosides. Please don't forget here. Let me show you so that it will be easy for you. See here. Now what are the 2 types of glycolipids which we have discussed? There are cerebrosides as well as gangliosides. Remember, the cerebrosides and gangliosides, they are further classified into different types, which we will study in detail in the biochemistry. But here I just want you to know one important point. This ganglioside, especially GM1 ganglioside, okay, there are different types, GM1, GM2, GM3. GM1 ganglioside, which is present on the cell membrane, it's acting as a receptor for cholera-toxin. Okay, see, we all know that cholera is caused by Vibrio cholera. It causes diarrhea, rice watery stools, all that thing. Now cholera produces cholera toxin. Now this cholera toxin enters into the cell with the help of a receptor called as GM1 ganglioside. Let me show you here so that it will be more easy for you to understand. See here, this organism what I am showing you, just take it as Vibrio cholera. Now this Vibrio cholera is producing a toxin called as cholera toxin. Now this cholera toxin is using this receptor, which is nothing but GM1 ganglioside. By binding to GM1 ganglioside, it enters into the cell and causes a cascade of events. Later will further cause diarrhea in the person. Okay, now that's the one important point I want you to know for your exams. Cholera toxin, please take it here. Cholera toxin uses GM1 ganglioside, okay, GM1 ganglioside as receptor, okay, for entry into the cell. Now after this, let's talk about the fluidity. Okay, this is a total different topic. Now, how fluid the cell membrane is? Yes, the fluidity of the cell membrane is because of lipids, there is no doubt. Okay, the lipids are arranged in bilayer and this is a fluid. Now, this fluidity is dependent on certain things or certain fats which are called as fatty acids. Guys, now let's discuss about a topic called as fluidity of cell membrane. Now, we all know that the fluids are nothing but the bilipid layer. Now this fluidity is determined by certain substances called as fatty acids. So whenever you take unsaturated fatty acids, they can affect the fluidity of the cell membrane. And saturated fatty acids, they can also affect the fluidity of the cell membrane. Now first let's discuss about the unsaturated fatty acids. Okay. What are the examples of unsaturated fatty acids, guys? 3 important unsaturated fatty acids which you should know for your entire career is linoleic acid and linolenic acid and arachidonic acid. Okay, so linoleic acid, and arachidonic acid, which you will study in your biochemistry, but just know here linoleic acid is omega 6 fatty acid and arachidonic acid is also omega 6 fatty acid, but linolenic acid is omega 3 fatty acid. Here examples of saturated fatty acids include palmitic acid and stearic acid. Now, what is the importance of these fatty acids in the topic of cell membrane, the fluidity of cell membrane is that, see, these fatty acids, unsaturated fatty acids, they are good for the body. Okay, unsaturated fatty acids, they are good for health. Okay, why we are calling they are good for health? It's because these unsaturated fatty acids increase the fluidity. Okay, increases the fluidity, which means the cell membrane, the lipid bilayer is now very much flexible. It's now easily going to release the substances and take the substances. Okay, cellular exchange is going to be very much easy. The fluidity of the cell membrane is going to be increased because of this unsaturated fatty acids like linolenic as well as arachidonic fatty acids. Now these fatty acids are very much high in concentration. If you want to know, just know this point. These linoleic as well as arachidonic acid, they are high in fish oil. Okay, fish oil is a very good source of these unsaturated fatty acids. Okay, now they increases the fluidity. But when I'm talking about saturated fatty acids, they are very bad for the health. Okay, why they are bad for the health? It's because they decreases the fluidity. Now what does I mean by decrease the fluidity? They makes the cell membrane more rigid. Okay, they makes the cell membrane more rigid. If the cell membranes are rigid, the cellular exchanges cannot happen. Cell cannot excrete the waste materials or it cannot take the materials from outside. So cell membrane is getting rigid, fluidity is getting decreases. So they are very much bad for the health and they are highly present in junk foods. Okay, junk foods are having richest source of this saturated fatty acids which are bad for the body. And these saturated fatty acids, the palmitic acid and stearic acid, they are also called as trans fats. Just remember guys, they are also called as trans fats. So whenever you are taking some product from outside, just look at the nutritional value. If you find trans fats in them, just try to avoid it, because trans fats like stearic acid, palmitic acid, they will increase the rigidity of the cell membrane, which is very much bad. They will decrease the fluidity, which is very, very bad. Now after discussing about the fluidity, let's talk about an important MCQ which came in exam. So fluidity buffer. Which substance is acting as a fluidity buffer in the cell membranes? What does it mean by fluidity buffer? For example, when a cell membrane is getting more and more rigid, now this substance will bring back the fluidity to normal, something like that. So the fluidity buffer in the cell membrane is maintained by cholesterol. Very important MCQ. Cholesterol in the cell membrane is acting as a fluidity buffer. Okay guys, now we have discussed all the different types of lipids. Now in the next video, let's continue this topic and discuss about different types of proteins which are present in the cell membrane.